Welcome to the Neurology Live Mind Moments podcast. Tune in to hear leaders in neurology sound off on topics that impact your clinical practice. I'm your host, Marco Meglio, editor with Neurology Live. In this episode, we sat down with Dr. Sharon Cohen, a behavioral neurologist and medical director of the Toronto Memory Program at the University of Toronto, who discussed an ongoing successful phase 1 study of Mivelsiran, the first investigational RNA therapeutic targeting amyloid precursor protein for Alzheimer disease. In the conversation, Cohen described the data presented at AAIC 2024, which included safety, efficacy, biomarker, and pharmacokinetic analyses. In addition, she spoke on the unique mechanism of action of this drug, the potential of RNA therapeutics, and how they would work with previously approved anti-amyloid medications. Let's dive in.
ALN-APP, and it now has a name, Mivelsiran. It is a small interfering RNA molecule. So it belongs to the class of RNA interference therapeutics. And this is an exciting way to target messenger RNA within the cell, in this case within neurons, and to shut off the production of the targeted protein or messenger RNA transcription. So in the case of ALN-APP, the target is amyloid precursor protein. And this is the parent protein that gives rise to all of the daughter amyloid proteins, A beta 40, A beta 42, and other A beta species extracellularly, as well as fragments intracellularly, like soluble APP alpha and beta. So to be able to go very upstream and target production as opposed to clearance of already formed amyloid, A beta 40 and 42, is very attractive. And you would think this maybe speaks to a prevention mechanism, but throughout the lifespan of someone with Alzheimer's, or throughout the duration they have the disease, they are continually forming more amyloid. And so if you can shut off that production, even if you have a drug that maybe clears the already formed amyloid, you're going to keep having more. So going upstream is very attractive to myself, where I'm very prevention minded. And we're starting to get some data from the first program. And this drug, ALN-APP, it's the first drug in this class of RNA silencing therapeutics where we're going within the brain. So we're delivering it intrathecally. So there are some drugs now, since 2018 onwards, there's been this new class of RNA silencing therapeutics targeting different organs, different diseases. The first one was in 2018 targeting peripheral neuropathy that had a genetic etiology, patisiran. And so that's how the market, and since then there have been, I think, at least 5 others. But with Mivelsiran, this is the first time we're introducing an siRNA directly into the nervous system. So pretty exciting to think we can go in the brain and shut off production of proteins that we think are involved in disease generation.
I'm curious, obviously it's a very, very cool mechanism of action, but, where are some of the barriers, I guess, with RNA therapeutics, and, why haven't we, I guess, had one of these beforehand? Or we're still learning more about Alzheimer's in general, but where are some of the barriers, I guess, to the development of RNA therapeutics, and how can we potentially overcome them with this therapy?
Yeah, so there are different ways that you can target messenger RNA, and there's a class of drug called antisense oligonucleotides that are also now having success at altering transcription and production of proteins from messenger RNA. And in the central nervous system, we have, as of last year, FDA approval of tofersen for a genetic type of ALS, familial SOD1 or SOD1 ALS. So that's also a way to target messenger RNA. With the RNA silencing drugs, it works a little bit different. We've got double-stranded DNA entering the cytoplasm of the neuron and being escorted to a naturally occurring messenger RNA catalyzing and degrading complex. It's called the RISC, or the RNA-induced silencing complex. We're actually taking advantage of a naturally occurring way of getting rid of excess or mutant RNA. And this double-stranded drug is very stable, but that's through some modifications over the years that the developers have learned to apply. So there's a carbon-16 attachment, if you will, to stabilize it. But once delivered to RISC, it's able to engage its targets. It becomes a single strand and is very accurately targeting the complementary messenger RNA sequence. So I think it's been, fairly rapid development since around, in the last decade, since this class of drug was described and developed. But getting it into the central nervous system and ensuring it's stable and doesn't have off-target effects has been, something that's taken a little time. When you look at the ASOs that get delivered intrathecally, they are single-stranded. Some of them act in the nucleus of the cell, some in the cytoplasm, but they are less long-acting and have more opportunity to have off-target effects because they have more modifications to aid their stability. So more links there that may be subject to disruption. So two different ways of modifying messenger RNA.
We're going to take a quick break from our conversation with Dr. Cohen to give you up to date on the latest developments in neurology. Welcome to the Neurology News Minute. Here's what you may have missed since our last episode. First, on August 9th, the FDA cleared an investigational new drug application for Adaptive Therapeutics cell therapy IDP-023 to be assessed in a phase 1 trial of patients with progressive multiple sclerosis. In the trial, investigators from Stanford University and the University of California, San Francisco will examine the biologic effects of IDP-023 using a translational program developed in collaboration by both universities. During the study, the participants with progressive MS will be given IDP-023 in combination with the FDA-approved anti-CD20 monoclonal antibody ocrelizumab. Days later, in a company update, Scholar Rock reported new positive 48-month data from its Phase 2 Topaz trial, with results showing continued and sustained motor function in patients with spinal muscular atrophy treated with investigational apitegromab, a muscle-targeting agent. Additionally, the company reported that it expects top-line data from its ongoing Phase 3 Sapphire trial of apitegromab in SMA in the fourth quarter of this year. At 48 months, a combination of 20 milligrams of apitegromab and nusinersen, a previously approved therapy for SMA, resulted in a change of 5.3 points in HFMSC in non-ambulatory patients aged 2 to 21. For those aged 2 to 12, the clinical effect was even greater, with changes of 6.4 points. And finally, on August 12th, the FDA granted approval to Medtronic's deep brain stimulation technology to be used while a patient is under general anesthesia or asleep in the context of treating Parkinson's disease or essential tremor. Typically, the standard established DBS procedure is based on awake surgery with intraoperative microelectrode recording associated with clinical testing to ensure good positioning of the lead. However, this procedure is sometimes perceived as uncomfortable for patients who are unable to tolerate awake surgery with extensive time off their medication. Ultimately, the FDA's decision to approve Medtronic's DBS for sleep purposes may provide greater treatment opportunities to a significant proportion of potential candidates for surgery. For more information about these stories, head to neurologylive.com, or you can check out the corresponding link in the show notes. And now, back to our conversation with Dr. Cohen. Let's talk a little bit about the actual phase 1 study itself. for our clinical audience, describe some of this phase one study, talk a little bit about some of the major points and the major takeaways. Obviously, still early phase on, but it's encouraging to see positive data. So talk a little bit about what we saw.
Sure. This is, it's such an interesting program. It's a phase one study looking at safety and tolerability as the primary outcome, but also pharmacodynamics, pharmacokinetics, and downstream exploratory biomarkers of disease. The population is young onset Alzheimer's disease. So it's not a phase one study in healthy controls. It's a phase one study in people who have a chance to benefit from knocking down or shutting off production of amyloid. And subjects had to have the onset of their disease under age 65. So we're dealing with a little bit younger population, not specifically looking for people with autosomal dominant Alzheimer's disease, but more young onset, thinking less comorbidity disease, more likely due to overproduction of amyloid than other comorbidities or other reasons. So that's the population. All of them confirmed to have amyloid through CSF or PET imaging, and they are at the MCI or mild dementia stage. So early AD in a young onset population. And it is a phase one study in two parts. Part A is a single ascending dose study. So what happens after you give one intrathecal dose and follow these patients for at least 6 months or out to 12 months, depending on the washout. So the part B, which we haven't reported on yet, is a multi-dose study where each individual will have more than one dose of drug. So what we presented at AAIC just was the last week, time flies by, was a cohort of patients now with a cutoff of, I think April was the date, 36 patients now with young onset Alzheimer's disease. Previously at CTAD and AAIC in 2023, we had reported smaller numbers and shorter durations of follow-up, but now we have 36 patients and we have follow-up out to 1 year. For the pharmacodynamic effects, of course, safety, full duration, and for the biomarkers of disease, A beta 40 and 42, we have results out to 6 months. So very exciting, and I can share the results. I mean, top line safety, since this is a phase one safety study, it is incredibly safe to deliver an RNA silencing drug targeting amyloid precursor protein. And how can that be? I mean, what does APP do? We know in neurodevelopment, it's pretty important, amyloid precursor protein in neurogenesis and synaptic formation and networking. So does it really do nothing in the adult brain? That's what you find out in a phase one study. Right now, we are seeing mostly mild to moderate adverse events related to lumbar puncture. And 3 patients were said to have adverse events related to the drug itself, but the same adverse events were also attributed by the investigator to the lumbar puncture at the same time and consisted of headache, neck pain, nausea. So sounds more like the lumbar puncture. There was one patient who had very transient on one blood test, mild lymphopenia, which resolved by the next blood test and no clinical symptoms. So again, that probably will disappear with a larger sample of patients and longer duration of safety monitoring. So the bottom line is we're seeing the kinds of side effects you would see in patients who have LPs. And you have to remember that in this phase one study, individuals are having 9 LPs within the first 6 months. So they have intrathecal dosing at day 0, they come back at day 2, they have an LP, they come back at day 15, and then month 1, 2, 3, 4, 5, 6. So 9 LPs, and it's really a testament to the fact that people are eager to have access to a therapeutic, even if it's in the investigational stage. So really, when you average the number of post-LP adverse events over the number of LPs that people are having, there's nothing extraordinary about it. So safe drug, and when we look at usual blood work and cardiograms and all the other kinds of safety monitoring, we're not seeing any signal that there's any concern. So if we move from the safety then, which we're pretty excited about, particularly when you think about anti-amyloid monoclonal antibodies and other classes of drug and the kinds of safety concerns that can occur. Here we're delivering something directly into the central nervous system and not having side effects that we can discern. So if we go to the pharmacodynamic markers, this is very exciting. Now we have results out to 1 year, and we see very robust, dramatic, and enduring knockdown of soluble APP alpha and beta. So these are the fragments that are sort of first off the chain when you catalytically, dispose of amyloid precursor protein from messenger RNA. So we're seeing in the order of about almost 75% knockdown maximum at 1 month. So that's early for amyloid soluble APP beta. So already by 1 month, you're seeing a real reduction, and it endures out to 1 year. Starts creeping back up, but there's still significant reduction in soluble APP beta and alpha out to 1 year. So that tells us there's target engagement. We're acting on the right protein. We're reducing production. We're not getting complete knockout. And this is a single ascending dose phase of the study. So there are more doses being tested that we haven't disclosed, and how much knockdown is the right amount is still to be determined. But we know that in a dose-dependent manner, we get this enduring and very substantial knockdown. Now, if we go to the more usual disease markers for Alzheimer's, A beta 40 and 42, we are also now seeing that we can impact those substantially. So we have data out to 6 months, and at month 1 for A beta 40, we're seeing about 65%, 66% reduction from baseline. And for A beta 42, it's about 50% at month 1. And then we have an enduring effect out to 6 months. So very, very hopeful data. Of course, the study's ongoing and more doses are being tested. And we haven't even begun to report on multi-dosing in Part B. But you can see that the profile emerging actually rather rapidly, that we have very accurate target engagement. We don't seem to have any off-target effects. We're getting the protein reduction that we're aiming for. And because of the enduring effect, there's the possibility that maybe this is a drug you could dose twice a year or something like that. Again, that's me talking. Alnylam's not putting that statement forward, but when you look at the durability of the response, that seems reasonable.
Dr. Cohen, as always, it's a pleasure to sit down and talk with you. I have one last question for you.
Sure.
We talk all the time about these new therapies, and now, it's very cool to see at AAIC that we can actually have discussions about real world data with some of these therapies, and the topics of these discussions are very, very much changing. So that leads me to my next question is, with this therapeutic, what is the potential and what does it look like if it were to be worked in with some of the other therapies that are already previously approved, talking specifically about lecanemab and donanemab? Could we potentially, obviously it's very early on right now, but could we potentially see synergistic effects? Could this therapy help out the other therapies? I don't know if there's any chance with this therapy potentially lowering some of the safety adverse events with the other ones. Just talk a little bit about some of the chemistry aspects of what this therapy could mean in comparison to some of the others, and whether we could see that combination approach.
It's so interesting to think about combinations now, because as you say, we are at a stage where we have some approved drugs and combination therapy is not such a far reach. So if we are dealing with a mechanism, as we are with the Mivelsiran, where we're shutting off production, we still have a lot of amyloid sitting in the brain. So it makes sense to use an anti-amyloid monoclonal antibody to clear the existing amyloid, both plaque, protofibrils in the case of lecanemab, oligomers, because those are toxic to the brain. But at the same time, knock down production so that you're really dealing with the disease in a more definitive way. And there's no indication from the safety data or from the mechanism of action that mivelsiran would cause ARIA because it's not clearing plaque. It's not clearing amyloid from the brain. It's shutting down production. So it works so differently from anything that would cause ARIA. It doesn't, to our knowledge, have an impact on inflammatory response. So unlike some of the drug combinations where you might think if you give a drug that dampens down microglia, then maybe you would have less ARIA. I think the best we could say with mivelsiran is you wouldn't, augment the ARIA effect, and you would you would reduce amyloid production so that, if you have a brain producing less amyloid, maybe that would lower ARIA, but ARIA is more from clearance of plaque. So I think, there's a whole host of combinations, and who knows, maybe an ASO targeting tau, there's one in development, along with an RNA silencing drug like mivelsiran to shut off production of amyloid. Depending how early you're going, maybe targeting amyloid and shutting it off is enough in prevention. But if you're at the early AD stage, you already have tau accumulating as well. So maybe that combination of shutting off amyloid and tau rather than the other combination, which is using monoclonal antibodies against amyloid and tau to clear pre-existing protein. So the future will be exciting. And I should say that mivelsiran has gone on to phase 2 development. We're involved in the phase 2 program in a little bit different population, CAA, so cerebral amyloid angiopathy. And these folks have an overproduction of, or at least injury in the brain from A beta 40, more so than A beta 42. But the rationale, if you can lower all daughter species of amyloid precursor protein, including A beta 40, that would hold for CAA as well.
Thanks for listening to this episode of the Neurology Live Mind Moments podcast. To support the show, be sure to rate, review, and subscribe wherever you listen to podcasts. And as always, for more neurology news and expert-driven content, visit neurologylive.com.